We get everybody back together here after the break. What a nice panel the last panel was. So thank you all very much. That was great. Our last panel of the evening is on adoption of behavioral and social sciences research findings and research and practice. We're very pleased to have Gehlert from National Cancer Institute moderate this panel. As you know, I think we've made some really nice strides in dissemination, implementation, research, and thank you. Nice strides in dissemination, implementation, research. And it's been people like Gehlert who have really done a really nice job of sort of moving that forward at the NIH and making sure we get that kind of work done. So, Gehlert, thank you.
Thanks, Bill, and thanks, everyone. So, since that slide has already been covered, some of you may have seen this, but I thought this was a good cartoon, sort of illustrating the charge of this panel. And you all have heard a lot of great talks about research and research infrastructure, but I think sort of the ultimate question is, what should we really do with all of this research? And that's, part of what we think about in implementation science. And this graphic, which many of you may have seen or not, illustrates what happens if we aren't thinking about what we should do with our research and we just sit back and publish, which is the usual thing, and do no more. So here you can see in this so-called publication pathway, on your left side, those are sort of the leaks in the pipe, the negative results, going from the research to submission, acceptance, publication, and also lack of numbers and inconsistent indexing, leading all the way from research to eventual implementation of whatever the findings are. And on the right side of the slide, you see sort of the rate of flow, so to speak. So what these researchers found when they reviewed 6 large areas of research, and estimated that it takes approximately 17 years to turn 14% of original research to the benefit of patient care. So as researchers, we really need to do a better job. So part of the problem is we assume if you build it, just because we generate evidence doesn't necessarily mean anybody wants to use it or needs it. And so thinking about beyond the evidence is only as good as how and whether people are going to adopt it. Whether practitioners are trained to use it, and of those practitioners who are trained to use it, whether they actually choose to use it. And whether of those who choose to use it, whether their patient populations actually benefit from it. So if you assume a 50% threshold for each step, which, as I understand in behavioral and social sciences research, is quite a large threshold, actually, and in implementation science even. And you even assume perfect access, adherence, dosage, and maintenance, you still only see a 6% benefit. So we really need to do a better job of really thinking about these things and thinking about the reach. And this is the RE-AIM framework, which some of you may be familiar with, but this is originally developed as an evaluation framework, but is often used by many researchers to think through what, the so what of their research, and if they are thinking about implementation, what are the dimensions that they need to be thinking about: the reach, the effectiveness, adoption, implementation, and maintenance. So you don't have to be doing dissemination and implementation science to be thinking about these things. This is a sort of translational research spectrum from basic science to dissemination, implementation studies. And basically the point I wanted to highlight here, and I think part of the charge of the panel, is whatever stage you're at, and I actually would have added another circle around that very first orange circle, but these are the kinds of things that we can be thinking about earlier on. And what you'll be hearing from the panelists is not only about their research, but ways in which they are thinking about adoption and some questions to consider. So with that, I'd like to introduce the panelists. So we will first be hearing from Dr. Sarah Gehlert. who is the E. Desmond Lee Professor of Racial and Ethnic Diversity at the Brown School, a professor in the Department of Surgery of the School of Medicine, and a scholar in Washington University's Institute of Public Health. Dr. Gehlert is the co-program leader of the Prevention and Control Program of the Alvin J. Siteman Cancer Center, and she's also the co-director of the Transdisciplinary Center on Energetics and Cancer, which at NCI we know as TREC. She will be talking about TREC, funded by the NCI, and efforts their team has made to enhance adoption of their research. So welcome, Sarah, and thank you.
Good afternoon. I'm going to talk about the efforts we've taken to speed translation. In my 20 minutes, I'm going to describe the TREC initiative. We've already heard that it's called Transdisciplinary Research in Energetics and Health, but we refer to it as TREC. And the TREC at Washington University linked transdisciplinary team science to translation in the Washington University TREC, outline a longitudinal survey of the growth of TREC and its transdisciplinary ties and its results, and then suggest mechanisms for increasing research capacity to move toward translation and articulate some of the lessons that we've learned. So, TREC is an NCI U54 mechanism with the mission of integrating diverse disciplines to find effective interventions across the lifespan to reduce the burden of obesity and cancer and improve population health. And I wanted to briefly show you the structure. It has 4 research projects, one at University of California, San Diego, one at the University of Pennsylvania, one at Harvard, ours at Washington University, and then a 5th site is the TREC Coordination Center, which is at the Fred Hutchinson Cancer Center. So each of these sites of the research sites, has about 4 research projects, and altogether there are over 116 investigators from 30 disciplines, and I've shown you about 17 of the 30 disciplines because they wouldn't all fit on the slide. And we're really talking about the second funding cycle of TREC. So I've got too many devices here. So WashU TREC we'll talk about first. So WashU TREC is transdisciplinary and translational research to accelerate the march from basic discoveries to population change. It's not really too ambitious, is it? And it's transdisciplinary research. We think it may involve direct interaction between projects and the feedback of results, and I'm going to illustrate that in a second. It might produce distinct scientific findings that accelerate translation by capitalizing on results from individual projects, and I'll try to illustrate what I mean. So, this is a little busy. I've reduced it to just 2 projects instead of 4 to make a point. And so you've got core resources. We had a dissemination and implementation core. And then here we've got 2 projects and the resources of those projects. And they're really interactions between the projects. So you've got interactions between the projects, but you've also got, importantly, the feedback of results from one project to the other project. And then we think that from the interactions and the feedback, you've got translation of individual projects, but you've also got something greater, which is the translation of the results from the center as a whole. And that's what I'm going to be talking about today. Okay, this is another way of looking at it. This is something I published a few years ago. So in the old way of doing things, we'll say, you might have policy studies, clinical studies, animal studies, epi studies, and really the knowledge was disseminated to those disciplines. What we've done is try to create an incubator here, and the incubator, mutual information and integration, fosters translation by focusing investigators on applications and increasing the likelihood of publishing in journals that cross-cut disciplines and really have a broader leadership. So we think that's the dissemination beauty of the effort. And this is the WashU Trek. So there's a lot to read here, but these are the 4 projects. And if we look over here, Project 1 was pretty much basic science. It was an animal model of transgenerational transmission of prostate proliferation in pups from a high-fat maternal diet during pregnancy. Project 2 was a longitudinal study of physical activity and prostate cancer outcomes among African American men that focused too on sexual and urinary function. And it was interesting, before we even began, when we met and were writing the application, Kelly Moley, who was the leader of the first project, planned on using female pups and looking at breast cancer, but because she met the people from Project 2, she switched it to male pups. She's in OBGYN. She really hadn't worked with male pups in the past. But that was an adjustment early on before we got the funding. Then Project 3 is a survey of employees to determine the role of workplace policy in shaping obesogenic behaviors. And Project 4 is run by our systems modeling lab, and it's model building of the social determinants of obesity in non-Hodgkin lymphoma and other cancers through stakeholder engagement. So these are the things that were going on, and as I told you, there was some interaction, but really there were separate entities at that point. So what we did to try to foster communication and collaboration among all the investigators and all the projects, because when you think about it, the challenge of collaboration occurs at 3 levels. The first is with investigators with any one project. Then you've got projects within a site or center, second challenge, and then you've got collaboration between the sites or the centers in the initiative. And you're trying to coordinate all that and maximize communication. So one thing that we did to foster communication and collaboration was bring in experts from the outside. And at first we thought they were just going to be giving us feedback. But what happened was that they really kept us focusing on the bigger picture. Because when you're working in a project, you're trying to deal with the day-to-day issues, and you're trying to get your specific aims met. And when we would bring in these experts from around the country, they would say, oh, look at the possibilities of collaboration among your projects and among your investigators. And that was invaluable. They could be dispassionate because they didn't have to deal with the day-to-day operations or the specific aims. We also included on this external advisory panel the director of the St. Louis County Department of Health, because we wanted her to keep our feet to the fire in terms of clinical applications, and she did. So one of the things we did at the very beginning was to bring all the investigators into the same room and basically lock the door for about 6 hours. And we had a room where you could write on the walls. So we brought in an external facilitator who's a cancer researcher, and she helped us understand how each project and each person's interests fit into a larger whole, which also was invaluable. So we came up with a shared model. And what we first came up with was that exposures at multiple levels of influence across the life course lead to inefficiencies in cancer prevention, diagnosis, and treatment, which leads to an increased burden of cancer in Missouri. And each investigator situated herself or himself in the model. So we've got epigenetics here, we've got medical oncology here, we've got urology here, we've got population epi here. And that was really valuable because we saw the bigger whole and every person knew where they fit into the picture. And we turned it a little bit to identify exposures at multiple levels of influence across the lifespan, produce efficiencies in cancer prevention, diagnosis, and treatment, and decrease the burden of cancer in Missouri. And you see I've added the 4 projects here. And again, everybody began to see where they fit in. And the thing is, you knew that for us to answer the bigger questions and really to get to where we wanted to be, which was at the bottom, we had to work together. Everybody's work was important. So we decided to investigate the Trek ties across the 5 sites, the 4 research sites in the coordination center, and this kind of fell to me, which was a good thing. What we did was develop a survey, and we surveyed 1 year into the Trek initiative and then every year after. So we had 4 surveys of the 116 plus investigators. It says 2011, 2013, which journal reviewers were quick to point out was more than a year, but it was actually 13 months. And what every investigator got a list of every other investigator at the sites, so all 116 starting out. And we asked them to indicate if they were currently working with that person, and I'm going to give you in what areas, and then if they'd ever worked with them. So we wanted to see if they had ties before or if they'd developed ties through Trek. And we asked them about whether they had worked with that person in a study or grant, co-authored publications, co-authored presentations, mentoring or training, committee or group work, and other. We had another category. And we used social network analysis to understand what was going on. Let's see. So, social network analysis holds that denser networks suggest faster propagation of ideas and information. And so we define density as the number of actual ties over the number of possible ties. So if you look here, that's not the pointer, each of these circles or dots is an investigator. And the lines between them are ties. And here we have a pretty dense network. Every investigator is pretty much, we call those nodes, communicating with every other investigator. Here's a less dense network because the communication is happening really through 2 individuals, so that's less desirable. And when we, this is just one scheme, but basically you can see we have categories of disciplines here. We put the 30-some into categories. And you can see how many investigators there were of each type over time. It didn't change that much. We sort of increased, I think, in epi, and we brought on some more statisticians, which is not surprising. The network size varied. It grew a little bit. We had, by the way, at least an 85% response rate each time. And you see, surprisingly perhaps, that the density didn't change that much. So we're going to get back to that. But I want to show you first the Horseshoe Trek. In 2011, which was 1 year in, remember, and then 13 months later in 2013. And again, we've color-coded the nodes. Each node is an investigator. And you can see this. I'll make it bigger in the next slides. And the number is the number of the investigator. And a bigger node here means that that person had more connections. They were connecting more individuals. They were bringing together 2 individuals. So in terms of self-disclosure, that big green dot is me, and the big blue dot is Graham Kolitz. So a year in, most of the connections were being made by me and Graham, the director and the co-director. You saw a little bit of a node here in a medicine doc who turned out to be the leader of the mouse, the animal model project. And by 13 months later, the nice thing was that more people were emerging as leaders. They were connecting people. So you want to be dense. You worry about people out on the periphery, especially this person only has one tie. And other people have, well, this one has one tie. So we were able, through this analysis, to figure out who was less engaged. And it turned out to be clinician researchers who were often off-site when they were seeing patients. So we were able to, this is Kelly Moley, the leader of Project One, and we were able to change things around so that Kelly really transmitted information out to these people that were on site less often. So that worked. So this was nice because it gave us a way of sort of changing things or correcting mid-course. So getting back to TREC as a whole. Remember, we didn't see much change in density over time, but when we began to look at TREC as a whole and what was going on between the sites, we saw something different. So here, the ratio of the percentage of cross-site ties was about 15.66%. And you can still see the 5 sites. You can sort of figure out the coordination center is where it should be, in the middle, coordinating. And here are the sites. We're Site C, and so it looks a little bit like what you saw in the last slide. And the discipline categories are color-coded. So things were operating. When the ties were through grants, publications, mentorship and training, committees, and the work groups that we put together that cross-cut sites. Well, as you go through time, in 13 months later, the percentage of cross-site ties had doubled, which surprised us. So here you began to, you're less able to see which side is which side unless you look at the little letters. You began to see still at some sites these investigators that were really kind of operating on their own. So that's 13 months later. This is 12 months after that, continuing to go up, and you really can't tell one site from another. We worried about this one site, not disclosing, in which most of the communication was through the director, which is, again, suboptimal. And then by 2015, you really had trouble telling one site from another. So the cross-site ties were going up, growing again. So what we found is that cross-site ties grew from 15% to 40%. Within-site ties stayed basically the same, 0.8086 to 0.82, actually went down a little bit. And the lesson was that investigators were beginning to leverage expertise and resources across sites over time. So we really, one side had more expertise in intervention research. We had Nola Proctor as our dissemination and implementation core leader. 2 of the sites, Harvard and WashU, had high-speed gene sequencers, and we began to really, really work across sites. The collaboration over time is really interesting. So you've got the years here, and you've got from grants, and that went up. Publications doubled, more than doubled. Presentations doubled. Mentorship went up. But interestingly, committees didn't change much. They went down, if anything, which made me happy personally. I didn't need any more committee meetings. And the lesson there seemed to be, once we thought about it, the committees serve as springboards for grant submissions, for co-authored publications, presentations, and mentorships. And once those ties are set up, it's really, you don't need the meetings as much. So decided to compare what was happening with TREC with another multi-site initiative funded by NCI and other institutes aimed at being transdisciplinary too. And I was a little bit surprised when I saw this because they only had one wave of data. We're the only It's very difficult to do this, and we're lucky to have 4 waves of data. But when you look at the cross-site ties 1 year in, so really this is 1 year in and this is 1 year in, we had twice as many cross-site ties. They had more within-site ties. So trying to figure that out, we finally discovered that the Centers for Population Health and Health Disparities involved community-based participatory research. It was a target, which may have focused them more locally rather than outwardly from centers, while TREC really focused outward to share these resources, the resources of the various universities. Another difference was that TREC had a coordination center, while the Centers for Population Health and Health Disparities didn't. And that really took over planning of scientific meetings. It established work groups, and probably most importantly, it helped develop cross-site developmental awards. And those cross-site developmental awards really incentivized the work across sites. So I think that was what was going on. This is something that Kara Hall did, who I think is in the back of the room. But she was showing the first 5 years of TREC. It was 4 different research sites and the transdisciplinary tobacco use research centers, and comparing them to R01 mechanisms. And they noted a real lag in the first 2 to 3 years of the project, while the team science was finding itself. We didn't have the same lag, so they had fewer than 10 publications after 1 year. We had 16, and then we also looked at the number of publications that were multidiscipline and the number that were that involved more than one site. And the number that involved more than one site really went up. These are the data for 2015 are incomplete. That's why, so don't even look at that, I'd say. So this was sort of surprising and heartening. And again, we're thinking that in multi-site, multidisciplinary, you're probably going to be in journals that really have a wider readership. We're looking into that now. So to conclude, I wanted to show you what I think the mechanisms were that made it turn out the way it did. So one thing that really helped us to maximize this collaboration and communication was the joint construction of the unified model by all investigators. That was worth the 6 hours. Then we really had a continual dialogue between projects with adjustment in design and measurement as needed. We talked constantly. We had internal and external advisory boards, which really made a difference, as I told you before. They could broaden our lens when we tended to sort of regress to the mean of looking at our own disciplines. Also, it was really important to track the collaboration and make the mid-course corrections. We spent a lot of time in terms of sustainability communicating with university administrators to change our culture. This is something the External Advisory Board, Bob Hyatt actually, urged us to do. So I marched to the chancellor's office and marched to the provost's office, and we got some changes made in tenure and promotion policy. And it allowed us to sustain the changes we'd made in the campus culture. Also, the incentives for knowledge transmission between investigators, projects, and sites through the developmental awards really made a difference. Lessons learned. We learned that good epidemiological methods need to be tied to mechanistic basic science studies, not to mention social and behavioral. And we needed to engage more basic scientists in understanding cancer-related changes, which they didn't. And to make the intervention research accountable to change population health. So as we heard, many intervention researchers have a shelf full of interventions published in journals, but not used in practice. So dissemination and implementation research can help us to understand and bring about population health changes, helping them to adapt evidence-based interventions in real-world settings. It mattered in Missouri, especially. So I would conclude that funding transdisciplinary team science is a way to increase research success and translation to improve population health for a given level of support. It occurs by increasing communication between investigators, projects, and sites, by increasing collaboration between investigators, projects, and sites, by generating new research questions, methods, and analyses. Coming together the basics, the animal model people and a political scientist came together and adapted Bayesian statistics to compare small groups of rodents. So we considered it a success if we came up with new research questions that hadn't existed before in any discipline, new methods, and new analyses. And it also extended the reach of publications and presentation. So I'd like to acknowledge these people that contributed to the work, and thank you very much.